Hi, I'm Kaylee Boyce, and I'm a senior at the University of North Georgia. My research that I'm going to discuss today mainly focuses on how warmer winter temperatures are affecting ornate box turtle hibernation and physiology. Ornate box turtles are organisms native to the Midwest and are ectotherms, which means they rely on their external environment to regulate their internal temperature and body metabolism. This feature of ornate box turtles, and really all ectotherms, is important due to the continuous increase of global temperatures. With rising temperatures, warmer winters are being observed, which means that higher temperatures are actually increasing body metabolism in ectotherms, as well as increasing hibernacular temperatures, as well as turtle metabolic rate. That means as metabolic rate increases in our innate box turtles, they may experience serious energy depletion when they're supposed to be storing energy for spring emergence, reproduction, and overall survival. If their overall survival is impacted due to warmer winters, fecundity and clutch size may eventually begin to decrease, damaging the life history of these ectothermic organisms. So what our study did to dive into the effects of these warmer winters is that we overwintered female turtles indoors from October 2017 to April 2018 at either 4 degrees Celsius or 10 degrees Celsius. Oxygen consumption and carbon dioxide production were measured and analyzed using EXPA data to go ahead and look at the differences during hibernation. Lactate concentrations were measured from blood samples taken during overwintering, as well as other blood metabolites. And our Q value was calculated, which is the ratio of carbon dioxide production to oxygen consumption. The RQ value changes based on what energy source is actually going to be used for metabolism. So an RQ value closer to 0.7 indicates more lipid breakdown, while an RQ closer to 1 indicates more glycogen breakdown. So if we go ahead and take a look at some of our preliminary results, we can see that turtles in the warm treatment had higher RQ values. This result was significant. If we looked at oxygen consumption and carbon dioxide production between the two treatments, neither differed, and these results were not significant. We also measured lactate concentrations, and the concentrations between the two treatments did not differ and were not significant. So if we go ahead and analyze these early results, the RQ values for the turtles in the warm treatment averaged out to around 1, which indicates that these turtles were metabolizing carbohydrates for energy. In contrast, RQ values of turtles in the cold treatment averaged to around 0.7, which indicates that these turtles were metabolizing lipids over carbohydrates for energy. This is important to our study because turtles actually have a large glycogen store. And if they are depleting their glycogen store in hibernation when they're supposed to be saving that energy up, they will be weaker and have less energy when they emerge in the spring. This is significant to us because it confirms that warmer winters may be affecting turtle metabolic rate. So what we're hoping to do with future directions of this research is analyze more blood metabolite levels and go ahead and look further into what else is factoring into the changes in metabolism being seen, as well as changes to physiology with these warmer temperatures. I'd like to thank UNG GURC, the UNG Biology Department, UNL Biology Department, and Sigma Xi for funding my research, as well as Dr. Nair and Dr. Bachman. Thank you.